Good morning, everyone, and thank you for tuning into this presentation today. My name is Prem Mohanty. I'm a life sciences product specialist and a product marketing manager at Benchling. I'm excited to talk about antibody discovery today and some of the critical considerations, the associated data management to really accelerate your R&D. Over the past several decades, antibody discovery has grown significantly in terms of complexity. Today, it includes a wide range of platform technologies and methods. Some of the specific drivers of this complexity are the fact that the antibody candidates being discovered are just much more diverse structurally. In addition to well-established monoclonal antibodies, we have a plethora of bispecific formats and antibody drug conjugates. Second, the processes for screening and discovering new antibodies are constantly changing. Scientists are constantly refining the discovery platforms they have in place to find better drug candidates faster. And finally, there is increasing reliance on lab automation to increase throughput, improve productivity, and achieve greater efficiencies. If we look at typical antibody discovery teams, we see several common challenges. An antigen design scientist is dealing with archaic in silico software tools that are highly manual and have low throughput. Additionally, capturing institutional knowledge at the sequence level is often not possible. A hybridoma screening scientist is dealing with complex lab automation hardware and software across the different screening steps and has to piece together information from these multiple sources. An analytical scientist is struggling with handoffs of data, samples, and context between teams, which makes it difficult to share learnings across program teams. As science becomes more complex, we are seeing teams become much more specialized to increase the depth and efficiency they can bring to each step in the R&D process. Each of these teams is producing not just more data, but more complex data. There are more contextualizations that need to be maintained as you move downstream in the process and across programs. There are more streams of data, from lab automation and analytical instruments to experiments being done by lab scientists. So the industry is looking towards digital solutions here to not just manage the volumes of data, but to bring order and structure to it. We think every company and scientific team deserves a solution that will accelerate and support your innovations. When you think about the ideal traits that any digital solution should have in R&D, there are really 4 core attributes to consider. First, it has to model the science you focus on, which means having the ability to build data models that fit your science. Second, it has to support the processes and workflows that your team performs and have the ability to scale, iterate, and evolve over time. Third, it has to empower users with data at every level, giving scientists insights about their experiments and team leaders data on their programs, so you can make both scientific and operational decisions quickly with complete and accurate data. Finally, it ensures that users will immediately recognize the value of the solution, see that it saves them time, and adopt it in a way that will keep them compliant and consistent with the rest of the R&D organization. Let's look at each of these attributes a little more closely. So what does it mean to be built for complex science? There has to be alignment between experimental workflow and the way that the underlying biological entities and samples are modeled. As an example, in the case of a hybridoma technology-based antibody discovery process, the science here generally involves antigen design to selecting and creating immunogens relevant to the target you are going after, followed by animal immunization in transgenic mouse to generate the antibody-producing B cells. Then you do cell fusion, selection, and screening to select the ideal clones. And then you characterize the clones produced. And finally, you might do antibody engineering to produce the actual antibodies of interest. While this is a very simplified view of the process, it's actually very complex to model this. Every hybridoma has a variety of entities associated with it, such as the antigen used, source animal, tissue sample, parent B cells, fusion cell line, and the antibody produced. Each of these entities have their own sequences, properties, and ontology. Every related sample has experimental results that need to be tied back to not just the sample, but also its underlying parts and sequences. So this is clearly a data modeling challenge, but it is also an operational challenge. since these also represent the day-to-day functions for lab scientists developing these hybridomas. When we think of preparing digital infrastructure for these kinds of challenges, we have to think about both the data management and structure as well as the operational considerations. The way that we think about this is to build applications that follow the operational workflows, from in silico design to experimental execution, but to make sure that the data capture flows into a structured data model. This means at any point in the process, you should be able to trace your work back to all the preceding steps and underlying sequences. And more importantly, for scientific decision support, the data can be readily queried across any dimension of the data model. This is a necessity to start building in automation and machine learning routines. So that's an example of how a very complex scientific process can be structured into a digital solution. But the hallmark of any good program is that the science will evolve over time. So what happens when things change? Digital solutions have to be adaptable. The IT world has moved away from monolith software and towards more agile approaches. But adaptable shouldn't always have to mean coding and application development. Many biopharma companies either don't have the resources to do this or want to use the resources they do have to focus on higher value operations. This is an illustrative example of part of a hybridoma screening process, and it reflects the constant iteration and evolution that these specialized teams see on a daily basis. So in an ideal world, the data model, the process, and the workflow should be configurable in a low to no code environment, so companies can iterate and evolve quickly without being resource intensive or overly dependent on their digital partners. One way we seek to achieve this is with a configurable data model that is editable right from an admin panel. So users can adjust the relationships and metadata on a particular entity quickly and without code. This also applies to process workflows where you can update iterative processes like the example you see here. Once you have the data model and process in place to capture structured data, the next consideration is decision making. A modern digital solution should enable its users to make data-driven decisions. Different members of an organization have different questions that they need to answer and have varying decision support needs. For a scientist, this means uncovering insights across their experiments so that they can design, execute, interpret, and optimize their studies more efficiently without tedious data aggregation and data cleanup. For example, a scientist might be looking for an appropriate cell line to plan their study. Now, instead of emailing other team members or going to separate inventory and ELN tools to look for this information, the scientist should be able to look up and compare characterization profiles and inventory data of multiple cell lines by querying a central data warehouse where the data is already standardized during data capture. At the level of a team manager, the questions that need to be addressed are at the process and team productivity level. Now, typically, this information is collated and visualized with Excel or other specialized process management tools with separate data entry and reporting workflows. A modern digital solution should be able to connect the experimental and process level information in a single system, such that managers can track team productivity and throughput easily and answer questions such as number of outstanding requests, assay turnaround time, and view the latest progress on related experiments. Finally, at the executive level, you want a high-level view of your entire pipeline, the ability to compare candidates to make go-no-go decisions and allocate R&D resources efficiently. For example, the executives of an antibody research organization might want to look at key data across leading hybridoma heads to identify the best candidates to support subsequent research programs. Ease of use is a final important attribute of a modern digital solution. Most software tools have a learning curve, and then because of poor user experience, a forgetting curve. Only if the solution is easy to use can it ensure that users not only get trained, but ramp usage and realize the full value of the solution. An easy-to-use solution allows users to record experiments quickly, tap into the records to uncover the most meaningful results, and finally aggregate and analyze results across studies from the same user interface. This is essential to drive performance, boost productivity, and improve data integrity. The biopharma industry has evolved tremendously over the past decade. We are proud to work with several leading antibody companies that have helped develop new advances. We have been able to scale with these global biopharma leaders as they built out their antibody portfolios. We are closely aligned with these leading innovators and work in partnership. This has helped build up our domain expertise in antibody discovery. So, how can we help antibody discovery teams specifically? For animal immunization, we provide collaborative study management tools that are integrated with sample tracking. This means you can track the entire lineage of your tissue samples and cell lines and access the complete experimental history with a few clicks. For antibody screening, we provide the ability to integrate your existing lab automation hardware and software with your experiments to provide full end-to-end traceability. For analytical characterization, we provide the tools to easily study, analyze, report, and compare characterization data across candidates, studies, and programs. To give you some more specific examples of how we can support animal immunization workflows, I wanted to talk about these three things. First, the Benchling notebook can help you create and manage all your animal study plans and protocols in one place. This can then be used as templates for running the actual experiments. This streamlines collaboration between scientists in authoring these protocols and actually executing them. Second, Benchling's registry and inventory solution enable you to map the relationships between all your samples and track the physical locations of these samples. As a result, you can easily track the lineage of a given hybridoma cell line and view all the related experiments and data from a single registry entry. Finally, with Benchling Insights, you can create dashboards to monitor all your studies. Because these dashboards are linked to the experiments, entities, and results directly, any changes are automatically reflected in the dynamic dashboards. This can help you plan and optimize resource allocation. For antibody screening, Benchling can help maximize overall efficiency of your workflows. Notebook templates can make it easy to set up the replicate runs and enforce your in-house screening SOPs. These notebook entries also help capture data in a structured manner, which helps with data analysis later. Second, we can integrate with your existing lab automation hardware and software to help capture results directly into your notebook and automatically link these results to the hits in the registry. This improves traceability and data integrity across all the screening steps. Finally, the in-platform data visualization tool can give you a high-level view of your entire process from start to finish. You can evaluate the throughput, turnaround times, the statuses of discrete steps of the screening workflow. This can all help you refine your process further or resolve bottlenecks quickly. For analytical characterization, Benchling can really help bring all the characterization data together automatically for easy analysis and interpretation. With our molecular biology application, you get access to a comprehensive suite of the latest in silico tools to analyze antibody sequences and sequence level features such as CDRs, framework regions, amino acid residues, and sequence liability sites in a high throughput manner. This cuts down the need to use a variety of different molecular biology tools, reduces manual effort, and boosts throughput to meet screening objectives. We can integrate with analytical instruments to automatically bring results directly into your notebook entries. This boosts productivity and improves data integrity because you no longer have to spend the time manually assimilating all these data from the disparate sources. With our in-platform visualization tools, scientists can create complex dashboards to analyze characterization results across studies, candidates, and programs. This saves scientists countless hours digging up data and manually putting together data for performing the same comparisons. Everything that I talked about just now is powered by the Benchling Life Sciences R&D Cloud, which is designed to give you a complete view of your R&D innovation. Benchling's applications span the R&D process from experimental design, in silico biology, sample tracking to process management. There are 7 applications that Benchling offers, but they are all offered from a single interface. This makes it easy for users to leverage what they need without leveraging the software. All of these applications are built on a common unified platform. They share a configurable data model that maps to each team's unique scientific and operational requirements. Benchling is built on a modern cloud infrastructure, fully managed by Benchling and offers state-of-the-art security and control. And we know that most companies have a rich ecosystem of instruments, software applications, and data stores that the R&D depends on. So we provide an open platform with a full set of REST APIs out of the box for and connectors for lab automation, as well as ways to move data onto and out of the platform. I can share an example of one of Benchling's customers. Agenus is a publicly listed clinical stage immuno-oncology company based out of the Boston area. Prior to using Benchling, they operated from a variety of point solutions. This made it difficult for scientists to find information and collaborate, especially across multiple R&D sites in the US and EU. Additionally, because of the complexity of their biologics workflows, it was difficult for them to keep track of their experiments and samples. They adopted the Benchling platform back in 2016 with the notebook, molecular biology, and registry applications. They have subsequently expanded to include the entire Benchling platform for their scientists. Their entire antibody research team can now collaborate better around a single unified platform with a complete view of their experiments, samples, and results. They can mine data across past experiments to optimize future studies. This is just one of the many examples on how Benchling customers can accelerate their R&D using Benchling. And you can find many more of these success stories on our website. Benchling's mission is to accelerate life science innovation for the betterment of humanity. We have built out a range of solutions for life sciences on a single platform and continue to invest aggressively in our product development teams to bring even more powerful and transformative features and products for our customers. Please reach out to me if you have any questions or would like to learn more about how we can support you and your teams.